For video number 3, we chose to answer question number 2 posed in Ellis class session number 1. Why would an individual show an increased blood-free fatty acid content and a decrease in skeletal muscle membrane GLUT4 content following a 48-hour high-fat diet? According to the data provided, those participants who ingested a high-fat diet experienced a constant increase in blood-free fatty acid to a point of 30% change, compared to those participants in the placebo group that experienced no change in blood-free fatty acid content whatsoever. Within the same group of participants, data suggests that over the same 48-hour period, the high-fat diet sample saw a 50% decrease in their skeletal muscle membrane GLUT4 content in comparison to the placebo group.
As a group, we hypothesized that the elevated levels of blood-free fatty acid concentration in participants due to an acute high-fat diet will result in more free fatty acid being translocated into the muscle cell, resulting in a higher concentration of ceramides and DAGs. We also expect to see an increase in TNF-alpha due to adipose cell hypertrophy. Together, these will inhibit the insulin receptor pathway, which will lead to a decrease in membrane GLUT4 proteins and ultimately lower insulin sensitivity. Now let's look at the information needed to understand our hypothesis. The insulin receptor activates the insulin receptor substrate, which then activates PI3K and AKT. Blood-free fatty acid enters the cell via CD36, where it becomes fatty acid acyl-CoA. There are 3 fates of fatty acid acyl-CoA, including reesterification, which results in an increase in DAGs that inhibit the insulin receptor substrate. The second fate of fatty acid acyl-CoA is sphingolipid formation, resulting in an increase in ceramides that inhibit AKT. The third fate is the oxidation of fatty acid acyl-CoA in the mitochondria. If there is no inhibition of the insulin receptor cascade from the reactive lipid species, AKT would phosphorylate AS160, allowing for the translocation of the glucose storage vesicle to the cell membrane. The release of RAC from IRS assists the movement of GLUT4 along the actin filaments.
The hypertrophy of adipose tissue results in a release of TNF-alpha, which inhibits IRS from releasing RAC and AKT from phosphorylating AS160. The mass we will be examining is GLUT4 membrane concentration, with inflow being AKT and outflow as glucose uptake. An increase in blood-free fatty acid concentration results in a decreased GLUT4 translocation to the membrane and ultimately a lower GLUT4 membrane concentration. This occurs due to an increased amount of DAGs, ceramides, and TNF-alpha, resulting in an inhibition of the insulin receptor cascade.
When researching this problem, we found an article by Lee and colleagues which looks at the different events underlying the development of insulin resistance following a short-term high-fat diet in mice. In particular, DAGs, ceramides, and TNF-alpha levels were examined. Although many studies investigate the long-term effects of high-fat diets with regards to insulin resistance, this study performed a short-term investigation in which mice were subjected to an oral glucose tolerance test following 3 days on a high-fat diet. As seen in this graph, compared to the control mice, which consumed a normal chow diet, the high-fat group grew to the insulin resistance within as little as 3 days. This trend continued for the 10-week study. This study also looked at whether macrophage release of inflammatory factors was associated with the development of insulin resistance. So clondronate liposomes were injected to decrease macrophage concentration. The black bars represents those injected with clondronate. Therefore, we are only concerned with the open bars, as they were the control mice that were fed different diets. In the control mice, elevated levels of triglycerides were observed in skeletal muscle cells. Along with elevated triglycerides, there is a greater formation of DAGs and ceramides from fatty acyl-CoA in the muscle cells. As previously mentioned, DAGs have been associated with the inhibition of IRS, and ceramides inhibit AKT. This suggests that an excessive fat uptake may lead to the inhibition of insulin receptor cascade, resulting in a decreased translocation of GLUT4 to the muscle membrane. In addition to the increase in fat species in muscle cells, adipose tissues were also affected. With the ingestion of a high-fat diet for 3 days, adipose cells were hypertrophied. As stated in lecture, larger fat cells release inflammatory factors such as IL-6 and TNF-alpha, both of which inhibit the insulin receptor pathway. Since IL-6 only affects liver cells, in our hypothesis we are only considering TNF-alpha levels, as this inhibits IRS and AS160 in muscle cells. Hypertrophied adipose cells also will release free fatty acids, which add to the high concentrations of fat in the blood and muscle cells. This study found that following just 3 days of a high-fat diet, mice had significantly higher levels of TNF-alpha released from adipose tissues. The authors determined that the elevated levels may also contribute to the insulin resistance observed in these mice. In conclusion, after being fed a high-fat diet for 72 hours, mice had significantly higher DAG, ceramide, and TNF-alpha concentrations. The elevation of these reactive lipid species and inflammatory factors inhibit different components of the insulin receptor cascade, which results in a decrease in GLUT4 translocation to the muscle membrane.